Lecture 7 can be a long and difficult one for students. I recommend that although you're following along with Socrative the first time you watch these videos, if you need to do so, go back and watch them again. There's also going to be additional videos which are posted that you can watch so that you get to hear someone else explain the material for you. The most important thing is to be active with how you interact. Although watching videos can be really helpful to you, and I do recommend that you use videos to help you get a good understanding of what's going on, you need to actively work with the material. That means not just reading your notes or watching videos, but doing something with the information. You can do new organizations of your notes. You can complete worksheets like recitation or do questions in mastering biology. But you need to be working with this material to help you be able to achieve our goals. I need students to be able to identify redox reactions and label the reactant that's oxidized and reduced, as well as identify the reducing agent and the oxidizing agent. You should be able to describe catabolic strategies and contrast the reactions, oxygen requirements, and ATP generations for each one of these catabolic strategies. You should associate dehydrogenase activity with the reduction of NADH and kinase activity with substrate level phosphorylation. Explain the importance of electron carriers and the reduction of NADH. Count and track carbons and phosphates as we go through these processes. Name important molecules and enzymes throughout each of these processes. Count products following each stage of cellular respiration and then apply this knowledge to calculating ATP production. Describe the players of the electron transport chain in chemiosmosis, as well as name and describe the forces generated by the activity of these players. And finally, evaluate how changes to these players influence ATP production. Go ahead and pause the video here so that you can try these 5 review questions before we get started. So what describes a metabolic pathway that releases energy by breaking down complex molecules? Well, there are 2 specific categories of metabolic pathways we've spoken about, anabolic and catabolic. The one that breaks down molecules to release energy is catabolic, and we're going to be looking at an example of catabolic pathways today. True or false, one enzyme can act at multiple stages of a pathway. You'll recall that enzymes are very specific. They have a very specific shape to them, which gives their function. This also means that their active site, the binding site for the molecule that they're going to be acting on, the reactants in the reaction, is also going to have a very particular shape, which means that it's only going to be able to act on very specific substrates. So this is false. You need to have a different enzyme for each stage of your metabolic pathway because each enzyme can only act on a very specific substrate. Now, when enzymes act as the end product of a particular process acting on an earlier stage, this is known as feedback inhibition. ATP is an adenosine triphosphate. It's a ribose nucleotide. Not only can it be incorporated into a ribonucleic acid as the A nucleotide in the chain, but it can also be used for energy, and the cell is able to use ATP to do work because ADP is going to be a lower free energy molecule than ATP. And so we can use energy coupling to introduce energy into otherwise endergonic processes. The terminal phosphate of ATP is removed by hydrolysis. This is what causes the high free energy molecule of ATP to become the smaller, lower free energy ADP and releases that energy to be able to do work in the cell. In catabolism, cells are going to utilize that chemical energy in a different way. Very large molecules like glucose have a lot of free energy. And so they have energy that when extracted can be used to do work. And part of that includes the ability to create ATP. Think of glucose like a gold bar. It's a big high free energy molecule, but you can't exactly go to your local ice cream store and buy an ice cream for $2 by handing them a gold bar. They're not going to be able to break that change for you. ATP is like $1 bills. You can take a large molecule of glucose, break it up into smaller $1 bills, which you can actually spend to be able to do things. Cellular respiration is an example of a catabolic pathway. And we're going to be looking today at how the process of cellular respiration gets from glucose to energy, because it's a very tightly regulated process. Anabolism, which is going to be our next lecture on lecture 8, we're going to use the example of photosynthesis. So tell me in Socrative, how does ATP provide chemical energy to chemical reactions? Hopefully you remembered that there's not going to be any energy stored in particular bonds or groups. There's no extra energy or extra electrons, and you can't just break a bond and magically get energy released. What's key here is to remember that this all has to do with free energy, and the fact that ATP has more free energy than ADP. Now we're going to take a look at another way in which we can measure that chemical potential energy of a molecule. This is known as the principle of redox. Chemical reactions that transfer electrons between reactants, these are oxidation and reduction reactions, or redox. In oxidation, a substance loses electrons. Since electrons are going to be a negative charge, we say that this molecule is going to be oxidized. The reverse is reduction, the gain of electrons. Since these are negatively charged, it results in a reduction of charge. I like to think of this numerically. If you were a neutral molecule, your charge is 0. But if you gain a negative electron, you would become -1. So that's why we call this process reduction. You might also sometimes remember that oxidation is loss and reduction is gain. That spells oil rig, O-I-L-R-I-G. Now, when molecules are oxidized, they become known as reducing agents, because in the process of this reaction, by becoming oxidized and losing electrons, they have to give them away to another molecule which is going to gain them, and so that agent becomes reduced. So these are reducing agents forcing the reduction of another molecule. By this same principle, molecules that are reduced are known as oxidizing agents, because in order for them to be reduced, they must oxidize something else. Now, this transfer of electrons is really great, and it's easy if you're able to measure that transfer directly. But oftentimes, these molecules aren't giving away an entire electron. It actually has more to do with how the electrons are shared. And so a molecule that has a more tightly held regulation of those electrons is going to be more negative than in a molecule where those electrons are less hogged. So what I'd like you to do is try the next 4 Socrative questions to identify bond types and see what impact that would have on different types of redox reactions. Once you've tried the next 4 questions on Socrative, come back and I'll go through the answers. So what kind of bond is carbon to hydrogen? Now I'm going to follow this up by showing you pictures of these molecules. Understand that you shouldn't have to look at pictures in order to be able to see them. But if you don't understand what's going on yet, these can be a nice visual. The electrons are shown as little blue dots. You'll note that in this example, those little blue dots are right between carbon and hydrogen. That's because carbon and hydrogen are both weakly electronegative, and since they are equally weakly electronegative, this is going to be a nonpolar covalent bond where the electrons are equally shared. What about carbon to oxygen? In this case, carbon is weakly electronegative and oxygen is highly electronegative, which means that oxygen is going to hog the electrons, resulting in a polar covalent bond. So how is the carbon atom different when it's sharing electrons with hydrogen versus oxygen? Well, carbon to hydrogen is going to be a nonpolar covalent bond. Carbon to oxygen is polar. And so you'll notice, and you can visually see here, that there's more hogging, there's more time that the electrons are spending around the carbon atom in a nonpolar bond and less time that those electrons are spending around the carbon in the polar bond. So the polarly bonded carbon actually has less electron density. Because it has less electrons, that means that that molecule is going to be less negative. So take a look at this reaction. If you take a look at methane here, methane, the carbon that's in the middle of the methane, this is the same molecules that you see here, is going to be transformed into carbon dioxide. So we have a carbon participating in non-polar covalent bonds, which is becoming a carbon participating in polar covalent bonds. What happens is that this molecule of carbon is being oxidized. When it's being oxidized, that is because it is losing that electron density because the oxygen is now hogging it. If you were to take a look here at the oxygen gas, which is oxygen to oxygen, is a non-polar covalent bond and the oxygens are being equally shared, is going to be reduced when it gets changed to water. In this case, because when it's polarly covalently bonded with hydrogen, that now that oxygen can hog the electrons, and so that hogging of electrons is reduction. If you take a look and apply this to the equation of cellular respiration, what we find is that the carbons in glucose are being oxidized to carbon dioxide, and that the oxygen is being reduced to water. Now, glucose is not going to directly give its electrons to oxygen. We're going to be tracking oxygens over a large series of processes, electrons over a large series of processes, because we're going to systematically be pulling that energy out, which is the final product of this reaction. Glucose is instead going to be oxidized a bit at a time, transferring a pair of electrons at a time via intermediates. So when we're tracking these electrons being taken from that molecule of glucose, they're being given to compounds like NAD+, which are known as electron carrier molecules. These electron carrier molecules will do as their name says and carry the electrons to the next stage of cellular respiration. It's at the final stage of cellular respiration where they can eventually give those electrons to oxygen. Enzymes that catalyze this pulling of electrons, that actually take the electrons from those highly reduced molecules, are known as dehydrogenases. D means to remove, and the hydrogens, so dehydrogenase, is that removal of hydrogens. ASE lets us know that this is an enzyme. So if you take a look at the bond here, we have a carbon bonded to a hydroxyl group. This is a polar covalent bond. What we have is dehydrogenase is capable of removing those two hydrogens from this reaction. Each hydrogen has one electron bound to that atom or associated with that atom. Each one of those electrons can now be donated to a molecule of NAD+, along with one of the hydrogens, forming the new bond. And then it leaves behind one hydrogen that gets a positive charge because it lost its electrons to the NADH. So here's an example of dehydrogenase. This is alcohol dehydrogenase. And alcohol dehydrogenase is an enzyme found in the human liver. It breaks ethanol down into acetaldehyde. Without alcohol dehydrogenase, beer and other alcoholic beverages would be toxic in the blood. It's necessary to be able to break these down. When we take NAD+, this molecule is becoming reduced because it's gaining the pair of electrons from the two hydrogens that it removed from the reactants using this dehydrogenase. So it's gaining two electrons as well as a hydrogen, and so it's becoming reduced. The ethanol becomes oxidized because the hydrogens were given away from that molecule. And this is all catalyzed by the enzyme alcohol dehydrogenase. So tell me, in Socrative, in this reaction, what would be the oxidizing agent? Well, oxidizing agents are going to be agents that force the oxidation of something else. Remember that oxidation is the loss of electrons. So if you're going to force something else to become oxidized, you need to become reduced. They lose electrons and you gain electrons. So oxidizing agents become reduced. In this reaction, NAD+ is going to be the molecule that gets reduced, and so this is also going to be the oxidizing agent.